well. And this is what it looks like after a storm, a picture from above. So yeah,
How much can they? How much are they resilient after a storm like that?
Usually they are very resilient. We go to the climate discussion again. Storms have always been there. And usually mangroves are able to bounce back. Of course they will be affected, and sometimes a cyclone is even good. You know, a forest with the completely closed canopy, if a big storm comes, the big trees will break and will die, and you will have conditions for new trees to come. But the problem with the cyclones is if they are too frequent and the forest does not have time to recover. Or if a cyclone comes and impacts in a forest that is already impacted, the resilience of that forest will be very low, so it will not be able to recover. So the problem again with climate change is not the fact that we have storms, is that they are more frequent and they are more severe. Okay, and we have a study that we did in the Savi Delta, for example, and we found that in more or less 14 years, mangrove was back in terms of extension. It was back to the previous condition, not in the same places. Some areas were cleared forever, like the mangrove never grew back, but it was able to expand to other areas.
And in terms of species composition?
All the same. We did not lose any species, and the mangrove is back. So we can say that all the ecological functions are back. A few changes in terms of distribution, but all the rest is back. So, but now imagine if before the 14 years you have another major cyclone, then you will have a problem. Yeah. Okay.
This last picture is Inhaca?
No, this is Vega. Lucy. He died. The famous he died.
So how is the
okay, I'll show you one picture later. I can say that more than 50% recovered, but is still in the process. So even after he died, it was a big cyclone, like 1,000 people died in Mozambique, Zimbabwe, and I forgot the other country. But a lot of people died. Impacts were really, really very high, but more than 50%, and that was in 2019. Today, 2023, more than 50% has recovered. So, yeah. Okay, so we do ecological assessment to see if the forest is, in which condition the forest is, and what kind of interventions do we need to do, if any, okay? And how do you start your assessment? First, you need your question, your research question, or what you want to monitor. Do I want to see changes in extent and distribution? Do I want to see the condition of trees? Do I want to see how much people are exploiting the mangroves? What are people exploiting in the mangroves? So you need to have a very clear objective of your research. Then you need to look at the tools that you have. Do I have, if I want to do change detection, for example, do I have tools? Do I have softwares? Do I have adequate protocols? What do I have? And then the resources, in terms of human resources, you need people who understand about mangroves, who know how to implement the protocols. You need time, you need money. Remember how much time we took to do 1 quadrant yesterday, and we were supposed to do 10. So imagine how much time we would need to do 10 quadrants. Time is a very important issue. You need money, not money, but money. You need field and laboratory equipment. So you combine all that information, and then you decide what method you are going to apply to do your assessment. Usually in the Rio region, Western Indian Ocean, Maputo, South Africa, Mozambique, Tanzania, Kenya, Madagascar, and the islands, this is what is commonly done. Mapping and change detection is very good to indicate occurrence, distribution, and changes over time. forest structural assessment is very good to tell us from the ground how is the mangrove doing, and you can do assessments in over different periods of time, and you can see if things are changing or not. But it's very time consuming, it's expensive as well. We also do biomass and carbon assessments, very related to the structural assessments, but you also find other studies that look at pollution, productivity, and restoration. In Maputo we do, in Mozambique we do the top 3, but also other few. And what do you do when you do this ecological assessment? For mapping and change detection, you usually take satellite images. I'm not going a lot in detail for this one because I'm not a GIS expert, but you usually take satellite images of different periods of time. The higher the resolution, the better the detail you will get, but to a smaller area. So you take images from different periods of time, from different years, you map, and then you compare what has changed from one period to another. It's a very good tool. It will tell you if the mangrove has expanded or reduced in terms of extension, but it tells you very little about the condition of the forest because you're only relying on the green area you have, the canopy cover that you have. So imagine if you have a forest with a high canopy and then, but something is happening beneath, you will not know. Sometimes the changes in the condition are not clearly reflected in the extension. We can use NDVI as a proxy for the condition, but you still miss a few things. It's very good, but it's always good if you can associate change detection with some ground truthing and see in the field what is going on. Change detection, for example, can tell you that you lost 300 hectares, but it will not tell you why. What happened. When you go to the field, you can know, you will see what is happening. Is it a disease? Is it people cutting? Was it an extreme event? You will be able to know that. So it's very important if you can associate mapping and change detection with the ground truthing. And here I just have an example. This was mapping and change detection in the Zambezi Delta. And just by looking at these numbers, we could tell a few things. For example, the overall change in the first thing, you see we have mangrove stable. This means no change, okay? Then you have gain. Remember we said that the mangrove is a very dynamic system. You lose and you gain. If there is no human interference, you may even have negative balance, losing more than what you gain. But in most of the cases, because we have human interference, that is what happens in most of the cases. In the Zambezi Delta, between, I think it was 2000 and 2013, we had gain, a large new area of mangrove, which is amazing because usually every place's mangrove is reducing. In this mapping, we had loss of mangrove, and you can see that was mostly at Matilde. You see that name there, over there, Matilde, and Malinga Pans. You can see these red spots, and this was Cyclonidae. We only know that it was Cyclonidae because we were able to go to the field. Otherwise, we see these huge spots, and we would not know what was going on there. And then you have gain, you have loss. Sometimes it's human, other times it's natural. And then you by subtracting what you lost and what you gained, you have the net change. At the end of the day, this is what will tell you what happened to your forest. You also do structural assessment. And in summary, what you do your desktop map first. Not necessarily change detection, but it can also be change detection. It will add information. You do a desktop map, and you build a system to choose. You can choose randomly, or you can select a significant representative number of sampling points. So here we describe, you do your desktop study, you identify the areas where you lost mangrove, where you gained mangrove, and where there was no change, just to make sure that you will have representative sampling points in all those conditions. Then, if possible, you consider NDVI. It's a proxy for the condition of the forest. And then, when you are also selecting your sampling points, it shouldn't be like that. But if you go to a very remote area, access is an issue. An area like the Zambezi Delta, access is an issue. There are areas where you cannot get there. So you consider the existence of human settlements, because you want to make sure that you will sample areas that are close to human settlements and areas that are far to human settlements to see what is the impact of people being there. Then you also want to look at roads to make sure that you will have roads or sea. You need to have some kind of access to your sampling point, and you can also look at other important features. So you make the grids on the top of your map, you distribute the sampling points, you number them or you name them, and then you create a system for randomly selecting your points. Then you see how many points for each of your categories that you have. You take the coordinates of each sampling point that you selected, and you go to the field. You put them on your GPS and you go to the field. Okay. It's good to make sure that your sampling points follow a transect. And what is a transect? Is a line perpendicular to the coastline. Why do you think it's important to have your points in a transect? Do you think you get any additional information? Did I say that?
Yeah.
Yes, why? Why sampling in a transect and not just randomly, and why the transect needs to be perpendicular to the coastline and not parallel? Do you understand what I mean by perpendicular and parallel?
Gradient.
The gradient, yes, the zonation that we spoke about yesterday, exactly. On the other side, you can also, if you don't care about the zonation, if that is not important for you, if you don't want to see any zonation pattern, you can also just pick random points. But for me, it's usually good to go in the transect. Do you have a question?
Me?
Yes? No? Okay. I see you. Okay, so this is one example. You can see a channel. This is not the sea. It's just that, it's actually the sea over there on the top and the channel here. So you have a transect. You see we named each quadrant. So to help us identify and go to the field. We discussed yesterday, you can do squares, or you can do the circular plots. Remember we talked about that yesterday. I prefer the squares, but circular plots are also fine. Which ones do you prefer? Interracial, you do interracial, yeah. I think in the mangrove is a different challenge, but it's okay. We discussed this yesterday, remember? So we will not waste our time here. Let's see if I have any additional information to give here. No, I think this is it.
We discussed about the teacher. Take the diameter, the circumference, like
Which one? Show me.
The ones that have tiltroots,
like this one is written 1.37,
I will ask Prof. Amina, but
it's just after the last
Amina, you see this one for Rhizophora? It says 1.37 above this root.
The top.
Not the top. It's not the top.
Why not?
It's this one. Not the top. It says 137 above this one.
I do the above the top
Yeah.
root.
The second, that's the last root, and you just
Yeah. Again, the most important thing is to stick to your protocol. If you do like that for the first one, you do for all of them. Yeah.
Because yesterday we had like
I remember.
the root
Yes.
here,
Yes.
you have to take one
Yeah,
for each.
yeah,
for each
yeah.
line.
It's complicated.
Yeah,
You have to climb it's not safe even, you know, it's not safe, okay? So one thing that we didn't do yesterday, and we will not have time to do that, is to collect soil samples to do carbon assessment, okay? Remember we said that the soil is the biggest carbon pool in the mangrove. So how do you collect soil samples? You use this core, you put it in the soil. I hope I can explain that in English. I hope my English is enough to explain that. So you have a metal core, is a structure like this one. You put it vertically to the soil and you bury it, doing this circular movement. Then you pull it up, and you have to remove the top layer, and then you section your sample in 4 different depths. The one that we use is usually for 1 meter deep, but you can go up to 2. You can go until where you can go, but usually the top 1 meter is the one that has more carbon. But you can also go to 2 meters. Some studies go up to 2 meters. But if you go 1 meter, most studies go 1 meter, and it's good enough. So you section your core into 4 interval depths. The first one is 0 to 50, then 50 to 30, 30 to 50, 50 to 100, which is 1 meter, 100 centimeters, not meters. And then you take the middle portion of each one of those sections. Yeah? And that is what you will take to your lab. You take to the lab, you take the wet and dry weight. Wet weight is the weight that you brought from the field. Dry weight you put in the oven for 48 hours at 60 degrees. And then you take up a small portion of your soil sample, and you put it in the oven, you burn it in the oven, and then you wait again. So the ashes represent the organic matter, and you weight the organic matter, you use a conversion factor, and you will have the amount of carbon. I will share with you the detailed protocol. That I think there's no point for us to go in all the details. I will share with you, and you will be able to see that. But in general, that is the process, okay? Okay. Remember, we took height and dBH in the mangrove, yeah? So height and dBH will be used to calculate all these things. You will calculate the basal area. The basal area tells you how much of tree area is in your forest. You measure the diameter, okay? And so with the diameter, you are going to have the area that each tree occupies where it stands. You see what I mean? So that is what your basal area will tell you. If you could put together all the trees in your forest, how much of the area that would take, okay? Then you also calculate these other numbers, the relative dominance, the relative density, and the frequency, and this will tell you the relationship between the species in the forest. Which species dominate, which species is more frequent, and which species appears more, okay? Then you will also calculate the importance value index, which is going to say which species is more important ecologically. You have Avicennia, you have Rhizophora, you have Cereus, but one of those species will be more important in terms of being more frequent, occurring more, higher density relative to the other species. And so that one is the species with the higher importance value. Then you have the complexity index, which combines a number of structural features. The number of species in the forest, all the other indicators that we had are relative to the species, but the last one is relative to the whole system, to the whole forest. So you take the number of species, you take the mean height, okay, you take the basal area, which is derived from the dBH, from the diameter, and then you take the mean density of your stands. You combine all of that, and you will know how complex your forest is. In general, more complex forests are in a better ecological condition and are in a position to provide more or better ecological services. So if you have a forest with a complexity index of 1 and you have a forest with a complexity index of 10, your forest with 10 is better in terms of providing ecological services than your forest with 1. Sometimes a low complexity index does not mean degradation. Suppose you have a forest with short trees, all intact. You have one species, for example, all your forest is intact, but they are short trees. And then on the other side, all trees intact, but high in terms of height and high density. None of them are impacted by human activity, but because the natural conditions of the forest are different, they will have different complexity indexes. So be careful not to say this forest is degraded because the complexity index is lower. You need to look at all those aspects in order to be able to say something accurate. Make sense? Yeah? Question?
I'm curiosity, why did you divide by 10 to the minus 5?
Because the number would be endless.
It was like, okay.
Yeah. Your densities, you can have a stand with 5,000 trees per hectare, yes. And then you multiply by 20 meters average height. This is a very tall forest. 20 meters is too much. Let's say 10 meters high, and then if the trunk is very big, your dBH will also, yeah, it's easier to work with the, yeah.
About the EVA. For calculate this index, we consider only the frequency of the species density.
Dominance, relative density, and relative frequency. For the EVA, yes.
Yeah, but in this, the only information that we have is a kind of distribution for the species. So maybe the species in a case can be, we can have not a lot of number of individuals of the species, but the ecological value of the species can be important also if we have not a lot of individuals.
Okay. This ecological value is, note that it's relative because it means one particular species relatively to the other species. It's not in terms of how much this one species do in the system. It's not like, for example, Avicennia has more room for birds or a wider shade, or it's not on that perspective, but it's in terms of what this species does relatively to other species.
Okay.
Always comparing to the other species that are there. So if you have, for example, in Saco, there is only, how many luminiferas did we see? Maybe 3. Avicennia marina will definitely have a higher importance value than luminifera, because it's relatively to the other species that occur in the same system.
Okay, I mean, these things
Yeah.
are relative.
Okay. Makes sense?
Yeah.
Okay. More questions? Okay. So for biomass and carbon, remember we said the other day there are allometric equations to calculate your biomass and then convert biomass to carbon. We have two, these are general equations for above ground and below ground biomass. You see this P, this specific wood density. They are numbers, they are standard numbers that were calculated and are used globally, but you can always use site-specific wood densities, which is very difficult to get because you need to basically kill a number of trees, and yeah, so we use the standard values that were calculated and everyone uses. So you can calculate your above ground biomass, your below ground biomass, and with the converting factor, usually it's 0.5, you can transform your biomass into carbon. So basically you calculate your biomass, then you will also have your carbon. For the soil, these are the calculations that you do. You calculate your bulk density, the dry weight over the volume. Remember I showed you that quarter that you used to extract the samples from the soil? That one is, the shape is of half of a cylinder. So you can know the volume of the portion of your sample that you take. You understand that? Yeah? Okay. And that is the volume that you are going to use to calculate the bulk density. You need the bulk density to calculate carbon density, and you need the the bulk density to calculate carbon concentration, and then the carbon concentration to calculate your soil carbon, right? You simply apply the formulas there. And then for the system, you calculate your total carbon, which is adding your above ground biomass. This is live biomass, trees and branches, whatever. Your below ground biomass, the roots, and for those two, you use the formulas that above ground carbon, sorry. I put things together. It says above ground, it should be carbon. Above ground carbon derived from biomass, below ground carbon also derived from biomass, and then you also add the soil carbon. Make sense? Okay, make sense for everyone, not for you?
Yeah, we can go back to the other.
Okay, you want to see the formulas?
Yeah. Why we don't have
height?
No. The
Altura?
we have the sand, gas, the oxygen,
This is just an example.
okay.
Yeah.
Okay.
But there are wood densities for all species, I think. It should be for all species. If you don't find for your species, there is an average for wood that you can use. Yeah?
And I missed, how do you transform the value of biomass in carbon?
Multiply 0.5. Yes. For biomass. In the soil, for your organic matter, you multiply by 0.45. It is also good if you can have your site-specific conversion factor, but it's difficult to get for all places. So we use usually 0.45.
Why is that the conversion factor? Like, what's the reason?
Because not all your biomass is carbon. You have other things there.
Okay, so it's just a general conversion factor. It's used always or specifically for one?
For other ecosystems, it's different. Remember the amount of organic matter, the amount of carbon in the, but no, I think you use it for everywhere. All soil, it's the same. Ne? It's a standard. Yeah. But for soil, you might, it shouldn't be standard because, but we use the same. Yeah, it's like for wood density, you see one you have 0.9, other you have 0.1, but yeah. Yeah, can we go? Okay. So now I have a few examples of ecological assessments that were done in Nampula, one in Nampula and the other one in Zambezi Delta. It's just for you to have an idea of how your report should look like, okay? So this one is on change detection. I already shared with you the change detection in the Zambezi Delta. It's the same pattern. You have your time zero, which is 2012, your time with which you want to compare 2022, you have loss area, you have gain area, and you have a net change. You also have a stable area. It's not here, I don't know why. Then you have your net change. You can see loss in every areas. When you put the balance, you lose mangroves everywhere. This is what a change detection map looks like. You see three colors: red for what you lost, dark green for what is new, and light green for what didn't change. Okay? Just an illustration. So when you talk about the structural parameters, you have your mean density. Remember, we had our sampling units. We counted the number of trees in our sampling units. That will tell us the density. Density is basically number of trees per unit of area. That is what we have in our first graph. In our second graph, we have the density per species. So you can tell in a forest which species has the high density. For example, you see Stereopstagal is gray, so you can tell this was the most important species in almost all sites. And then you have Chrysophora mucronata, which is the, first Avicennia. The dark blue, you see Avicennia here, it's also very important in this system. Chrysophora mucronata, not so common, okay? Chirocarpus, even less, the last one, the top blue, okay? So this is what your graph will look like. Another thing that you can do when you take your diameter of breast height, you can do the diameter distribution size classes. You make size classes for your diameter, and then you see how many, in terms of density, how many trees fit into each size class. What is that important? Young forest will have this kind of distribution following inverted J. You see the letter J? It will do the other way. What you have here. What you have, for example, the top one, okay? And what does this tell you? If you don't have this type of distribution, usually it means that something is wrong in your forest. It's expected to have larger densities of the small trees, and then as the size of your tree grows, you will have less and less individuals. If you don't have that shape, usually it means that something happened. And it could be either cutting, people maybe cut and they have a preference for a specific size class. It could be maybe an extreme event, storms. They can be selective in terms of the species that they will target. For example, Idai in Beira targeted the tallest trees. So you go to the forest and you see all tall trees fell. Makes sense, né? With the wind, the effect of the wind. The shorter trees were protected by the physical barrier. They were more, it's closer. But the ones who were higher, you see, imagine a bunch of kids and then there is someone high and then wind comes. Of course, the tallest are also less stable and they will face more wind, so they fall. So if you don't find the inverted J curve, pay attention. Maybe something is wrong with your forest. So you have to look more carefully and see what is going on. There is a disturbance. You need to look more carefully what is going on. And we have a number of forests with that situation here. I had one really bad example. Okay, but you can see, for example, you see that first one? You only have 3 size classes. And you see this last one, you have all size classes, but you have this one is missing. It could be that this one is being highly targeted. You need to go and see what of course your data, by this time, your data will be able to tell you what is going on. And note that we have also this graph parallel to this one, showing us the size class distribution for the stumps. So you can compare this one with this one and see if the missing size class is here. If the missing size class is here, then you know I have a problem with logging. If it's not there, then maybe it's natural or maybe it's mortality or whatever. But at least now you have a hint. Make sense?
Yes.
Okay. This is another thing that you can do. You plot all your trees. Here you have your height against your DBH, and this gives you a sense of what your forest looks like. You can easily tell, for example, that this is a very young forest. Your DBH does not reach 20. This is a more mature forest, but maybe the density is lower, which makes sense in mature forest. When your forest gets mature, your trees go taller, the babies disappear, so your density tends to be lower. But it is a good forest. It's healthy, it's a mature forest. You can look at this one. I would be worried with this forest, but maybe it's also a very young forest. Why? You see the DBH changes a lot, but the height doesn't change so much. So probably this is a dwarf forest. It doesn't go, it's not very tall. The trees cannot grow very high. So I would look carefully and see what is going on here. We'll double check with the stumps and with the conservation. So this is just to give you a sense, a feeling of how your forest looks like. Then here we have the complexity index. You can tell, you see the numbers? 1.9, now add 5. Remember we divided by 10, now add 5 zeros and see what you have. Not easy to manage. But here you can easily tell what forest is probably in a better condition, delivering better services. You see the complexity index of 5.1 for Nantaka, then you have 4.4 for Lumbu, and 4.7 for Lunga. These are your best forests. And in fact, the other data also told us that. Keep in mind Nantaka, Lumbu, and Lunga. Okay? And let's see the worst forest. It's Cabaceira, né? Okay. I want you also to look at the variation in DBH in the basal area, which is a reflection of the DBH. If you have a high DBH, average DBH, your basal area will also be high. So pay attention at your DBH, your height, your density, and the number of species, and see how these numbers affect the complexity index. Let me take as an example, okay, we can take Lumbu and another one with a similar, okay, yeah, we can take Lumbu and Lunga. Lumbu, this is the DBH, height 0.95.
That's not the height.
Oh, it's a basal area, thank you. The height is 2.1, the density is 3,000, right? Now let's see, Lunga is similar, 3.7, and you had 2.1, okay? But look at the density, much lower. You see, the density is much lower. So what changed? What?
The trees are less dense.
The what?
The trees are taller, but they are less dense.
So when you see your complexity index, it's always good to look not only at the number, but look at also how these other parameters change. Remember, I told you that a low complexity index does not mean necessarily degradation. Sometimes you might have, that would be, yeah. You remember if an impact comes, for example, a storm, and a lot of trees die, you have new trees coming. And if it's a young forest, the density tends to be high. So you compare that forest with a mature forest, your density will be what? Lower. Remember that? But you will also expect to have a high complexity index in that mature forest. Why? Because your basal area and your height will be big. So when you look at those numbers, always go back and see what is changing. If you are comparing two sides, okay, this forest is more complex than that other forest, but let me see what is happening in each forest. Make sense?
Okay.
Mm-hmm.
In DBH, if the first number meaning the DBH mensuration, what means the other number?
Standard deviation.
Okay.
And that is a very good question. The standard deviation, you have your average, okay? The average is what? You sum all your DBHs, right? And divide by the number of trees that you measured, right? But so maybe your average is 5, but some trees had 10 others had 2, for example. So the standard variation, standard error or standard variation will tell you what is the size of this variation. And if you have a high standard deviation, it means that your forest is very heterogeneous. You have trees with 5 centimeters dBH, and then you have other trees with 10 or 22. So your forest is not uniform. But if your standard deviation is smaller, it means that your forest looks alike, and every tree that you pick is representative to the other trees. If your standard deviation is high, that representativeness is not so good. Make sense? Okay. The other thing that we have, remember we were looking at the trunks and saying if it's straight, semi-straight, or crooked? Yeah. We can show that in 2 ways. The first one, in the first graph, we have in terms of density, and you can see this is an impact that all these forests have a high degree of impact, even though some are better than the others. All of them have a high degree of impact. Now look at the quality one, straight poles. Some sites don't even have straight poles. It's either semi-straight or crooked. Remember, we also had stumps for all these sides. And this is also 2 graphs that you can compare. How many of your crooked poles you have and how many of stumps you have. If the number of crooked poles and the number of stumps go together, probably it means that selective logging, because people will choose the straight poles, right? Yeah. So it means that selective logging is playing an important role in the forest structure. You have crooked poles also because people are cutting a lot. But sometimes you will have crooked poles and no stumps. So it means it's a natural characteristic of your forest. Again, don't look at crooked poles, a forest dominated by crooked poles, and say it's a degraded forest. It might not be. Sometimes it's because of the environmental condition we spoke about the other day. If salinity is high, the trees will not go tall. Now you look at this other place, it's the Zambezi Delta. It's a low impact area, but it's a very remote area, so it's low impact, but also the environmental conditions are much better. You have big rivers bringing a lot of fresh water and a lot of sediment, and again, it's a closed canopy. Remember what we said about closed canopies? Trees will compete for light, they will grow faster and taller. This I have never seen in my life. A forest dominated by straight poles. Have you ever seen? This was my first time. I have never seen, but it's because of those combined factors. Very good ecological characteristics and low impact. Yeah? I'm talking too much, ne? I see I'm looking at the watch. Remember, we were also talking about, yesterday we also assessed the forests in terms of cutting level. This is what you get. You can do again by density or by percentage, and this says a lot of how the forest is being managed, okay? Forests that are poorly managed will have a lot of stumps. Forests with some degree of knowledge will have more of partially cut, you know, because people know I cannot chop down the whole tree. So they go for branches, not for the whole tree. But if human pressure is very high, they will either cut severely, more than 50% of the branches, or the whole tree. So you can read all of that from these graphs. And again, we can see here, which side do you think is in a worse condition in terms of conservation? The first one, fungo.
No, sorry. The second graph.
The second one. This. Okay. If you want to compare this and this, or you want to compare the sides, this one is low impact. But if you look at each side, it's different. Like for example, you look at Malingapans, this place, you almost don't have it's almost everything is intact. You have dieback. This is the area that was affected by the cyclone. Remember the first map that I showed you with the big yellow, red dot is this side. So you see a lot of natural mortality. You see why is it important to say how the trees, if it's a stump or if it's natural mortality. This graph tells me that something is wrong in this place. I see a lot of trees that die naturally, and I don't see in the other sides. So if I don't have a change detection map and I'm just looking at this graph, I know this is either a disease or some climatic event or whatever, because I have a lot of trees that die naturally. But if I go to this place, Pambari, if I go to Hospital and Amarelo, I can say there is a lot of logging here, because they have, particularly Pambari, so many stumps, more than 50% of stumps in this site. If I want to intervene, where should I go? I go for this place, and this place. Clearly, they need more intervention. Education, alternatives, source of woods, whatever. But those would be my priorities. Yeah. And we can do the same assessment for the other sites. Usually, the forests will have more intact trees than any other thing, because remember, logging is selective, okay? And usually, in most of the forests, you have less of straight poles. But still, so having a high number of intact trees, it's not an indicator of low pressure. But per se, it's not conclusive. It can be that you cannot look only at intact and say, okay, 50% of the trees are intact. What you need to do, combine your information again with the quality of your poles, okay? Makes sense? You see, okay, if you see that all your forest has crooked poles, definitely most of them will be intact. Né? So these two information, you need to analyze them combined. One will inform more to the other. Make sense? Okay, I think this is my last, no, I still have two. This one is for regeneration. You have to look at regeneration to see if your forest is sustainable in the long term. And by sustainable, I mean is the population sustainable in the long term? Is the population producing enough babies for it to sustain with time? We expect a sustainable forest to have a proportion of 6 to 3 to 1, and I'm counting the babies up to 40, the children up to 150, and the youth up to 3 meters. Remember what we did yesterday? Yeah. So the proportion between them, the ratio, not proportion, the ratio between them should be 6 to 3 to 1. A forest will produce lots and lots and lots of babies. Naturally, the majority of them will die. That is how it happens. You know, even with other living beings, even with fish, they produce a lot of eggs, but the large majority will die. The same thing happens in the forest. For those class 2, also many of them will die. It's natural part of the forest. So if you find this proportion 6 to 3 to 1, your forest is okay. Usually that is not the case. But, for example, you look at, we don't have such case here, but let's look at the first place, fungo. You see fungo? You have 10 to 1 to 0. I'm very concerned. All the 10, out of 10, 9 died. The environment is really bad. And the one that lived also died. So we don't have youth in our population. This is a very big red light. So I have to go back to my forest, monitor over time, and understand if this is a situation that happened just this time that I'm sampling, or if it's something recurrent. If it's happening time and time again, then we need to intervene. We need to see what is happening, what is the stress that the forest is going through. You can also have a situation where you have your regeneration class 1 is lower than your regeneration class 2. You see what I mean? So instead of 6, 3, 1, you have, for example, 3, 6, and whatever other number. That is also a red line, a red sign. It means something is happening with your babies. Probably the children are from a previous season, not from this reproductive season, are from a previous season, and your forest is not producing enough babies. So again, you have to monitor over 2 years or 3 years, see what is going on, and then if the situation remains, you need to intervene. Okay? So this is the kind of information that your regeneration potential will tell you. See Bairro Hospital, this one. It's the same situation as 10-1-0. You have lots of babies, okay? Then you have regeneration 2, and you don't have 3. If you paid attention in the previous figures, Hospital was one of the worst areas. Lot of cutting, a lot of cutting pressure. So it means that human impact is already affecting the survivalness of the forest. Definitely, we need to intervene at Hospital, according to the data that we collected. Yeah? Question?
So, es para repetir en português.
Ok.
Yeah, so para...
O rácio entre as tuas classes, lembras que nós contamos o número de indivíduos para a classe 1, 2 e 3, não é? O rácio dessas 3 classes tem de ser 6 para 3 para 1. É raro tu encontrar esses números assim, iguaizinhos, né? Mas tens de ter muitos da tua primeira classe e à medida que vais caminhando para a terceira classe o número vai reduzindo. Quando tu tens um cenário diferente desse, significa que alguma coisa passa-se na tua floresta e tu tens de prestar mais atenção. Monitorar por 2 ou 3 anos, ver se a situação se repete. Se a situação se repetir, então precisas de intervir. Demos 2 exemplos. O exemplo do fungo, onde tu tens 10 para 1 para 0. Lembra-te que tu tinhas 3 para 6, morre metade, né? 6 para 3, morre metade. No teu 10 para 1, morreram 9, muito acima da metade. Então, alguma coisa está a matar os teus bebés, não está a deixar eles serem crianças. E depois as crianças não conseguem ser jovens. Se esta situação se repetir por 2 ou 3 anos, a tua floresta é um sinal claro de que a tua floresta está em problemas. A sustentabilidade dela a longo prazo está comprometida. Então vais ter que fazer algum tipo de intervenção. Depois demos outro exemplo do hospital, que é similar. Tens muitos bebés, tens crianças, não tens nenhum adulto. E relacionamos os números do hospital com as tabelas que vimos lá atrás. Tinha muito corte. Então está claro que o corte está a afetar o padrão de reprodução da tua floresta. Vais ter de fazer algum tipo de intervenção. Yeah? Ok?
Também também não entendendo um pouco. Botar para o.
Queres que eu volte? Aqui?
Na integração de aqui, sal a sal, em termos de condição,
sal a sal, este aqui?
Sim. Não sei se estou a fazer confusão com as cores, mas várias estarem em melhores condições.
Do que?
Comparando com as outras.
Sim. Este gráfico mostra isso. Tu tens muitas intactas.
Sim.
So he's saying that it looks like Sawasawa is in a better condition than the other forests. You have a lot of intact, tens muitas intactas. You have a few partially cut, but look at the stumps. There are so many stumps. Tens muitos secos.
Fiz essa pergunta porque o slide seguinte.
Aqui?
Sim, este.
Sawasawa. Pronto, está aqui. Só está a dizer 10, 1, 0.
Sim.
You have problems here.
Sim, você vê porque anteriormente a professora disse que não basta só ter uma percentagem elevada Por exemplo, aqui diz que a floresta é Inhaca.
Isso.
É preciso ter em conta outros elementos. Foi preciso tentar perceber o produto.
So
uma percentagem que diga que ela é intacta, mas quando vamos fazer uma renovação, percebe-se que é algo.
Provavelmente, Sawa tem muitas árvores tortas. Por isso é que, bom, tem várias coisas que podem combinar aqui. Posso falar inglês? I think. Ou tanto já perceber? Are you all understanding? But Amina also wants to get it. So probably you need to think that a few things may happen in Sawa. The first thing, the average stand density is high, independent of cut or not. And usually, when the average density is high, means young forest, usually shrubby, usually crooked, usually. You have to make sure you go back to the other data and see if that is really what is happening. But usually, high density, young forest, crooked, or if not crooked, not so high. So people will not have a lot of interest cutting those trees. They are not valuable. That is the first thing. But again, you see that the density of stumps is really high. One other tool that you can use to help you is you do the density, the graph with the densities, then you do the graph with the percentages. It will add a little more perspective, because here with the densities, you tend to compare with the other sites, but if you do the percentage, you will know which, what portion of your whole forest the stumps is. You can tell easily that the majority is intact, but what is the percentage of your stumps is high. And with this, you cannot tell for sure. So if you do with the percentage, you will have a clearer view. I usually like to have them both. These are for different sites. But for the same site, you do the percentage and you also do the density. But Sawa, it's not a good place. Yeah? You, again, check this one with your density of stumps. Remember, we have the size distribution for density for stumps as well. You also check it against your stumps. Ficou claro?
Yeah.
Okay? Okay, so the last one is the total carbon. This is no news. You know how to calculate biomass. You just convert biomass into carbon. And what I wanted to show you here, you see the amount of carbon in the systems is different. Remember, the Zambezi Delta is that one with the high input of fresh water, tall trees and whatever, so you have more carbon than in this other also impacted. And you can, if the structure of the forest is affected, you can also expect less carbon. But remember, the biggest carbon pool in the mangrove is the soil. So if the impact did not affect significantly the soil, you can still have a lot of carbon in your forest. So don't look at a degraded forest and say there is no carbon here. No. It could be in the soil and a lot of carbon, as you can see here, okay? And finally, you can also pair your structural ecological data with socioeconomic data, which is what you did today. This is just to show you the kind of information that you can collect and will help you understand what is going on physically in the forest. You can ask people how they explore the mangroves. And they will tell you, we collect crabs, we collect wood. Okay, this matches with my results. Or if they tell you that they don't collect wood, and your forest is in a good condition, it also matches your results, okay? And also by knowing the uses that people do in the mangroves is good for you to make the management recommendations. If people, you see so many options, so many resources are exploited. Maybe you can propose or you can introduce new ways, different ways to explore mangrove resources. For example, you see in some places they don't collect honey. So maybe you can present them the idea. Can you stop chopping down mangroves and do honey production? So if you know what people do, you have wider options to propose your management recommendation, and of course, you will also understand better what is going on. It's also good to ask people how they perceive the condition of the mangrove, if they think the mangrove is okay or degraded or whatever, and what they think are the causes. Did you ask that question today? No. It doesn't matter, but it's really interesting to see the community perspective. Remember, Prof. Amina talked about the local knowledge, the indigenous knowledge. In my experience, they know. Sometimes the knowledge is not systematized, it's not systematic, it's not fancy, I would say like that. It's not fancy. Sometimes there are some connections that they don't make, but they know. They know that a storm came, they know that people cut this place or that place, they know that the fish disappeared, they know. So it's always good to ask, even if it's not like a household or asking everyone. If you find someone inside the forest, you ask, you chat a little bit, and then you make key questions, you ask key questions, will help you understand what is going on in the forest. And people, it's also good to ask them, to ask the community what management strategies they would recommend. This was, we asked the community if they think that creating a community conservation area was a good idea or not. And a lot of people said, no, it's a good idea, yes, we need that. Others said, I don't know, and usually when they say I don't know, it means it's a bad idea, we don't want that. But yeah, so you can also use that to sense how people would receive that idea. If everyone says, I don't know, or it's a bad idea, then you know it's not good to approach them and say, let's make a conservation area. But if they are open, then you know you have a flow for discussion. Sometimes they say it's a good idea, but they don't understand the concept of conservation area. But at least you know you have a flow to start your discussion. So based on all the data you collect, you make the management measures. You can see if your exploitation is at a good level, or if you need to intervene, you need to preserve, you need to restore. It's also an option. You can tell exactly which areas would need intervention. You can tell if people have preference for one or other species. So the forest is good, but maybe this species, if we keep cutting like this, it will be endangered. So we need to do something about it. So, and you also, of course, look at the potential for carbon projects, which Prof. Amina will tell us about. And that's it for me. Questions? Do you have more questions? You have a question or a comment?
No, I was thinking maybe after.
Okay. But you can think aloud.
Is there like a paper or report about Wisdom Purake study that can be interesting?
Yes. I just don't know if I can share.
Okay. So
Because you are working for Oikos.
No,
No?
George is
George.
I'm probably going back to work with IDE,
Okay.
International Development Enterprises.
You are going to do one of the restorations over there.
They have a big project in Ampula.
Financed by BAF. BAF, by Blue Action Fund.
I
Financed
think
by the
so,
Blue Action
and
Fund.
they're working also with together.
I know.
And the founders are the USA.
Okay. I would advise you to contact WCS. The results that I just showed you, yes, are in a report. I would advise you to contact WCS and ask them for the report. They are the ones who should share, not me.
Okay, thank you.
Okay. More questions?
Yeah.
Sim.
Sim, não, professora, eu estou muito interessado neste conteúdo, quero usar ele para uma tese de mestrado. Okay? Então, o que eu gostaria de saber, professora, é que quando nós fazemos um assessment, a uma questão como o mangá, temos que fazer a mensuração de todos esses parâmetros, por exemplo, o nível de corte, o carbono e o estado de regeneração. Quantas coisas é que nós devemos fazer para termos um resultado assim que nos faz tomar alguma decisão?
Did you understand his question? Here is the answer.
Yeah.
What is your question? What do you want? You need to know what you want. If you want a general picture, if you want to know everything, You can do this and more. This is not all. You can do more. You can look at fauna, for example, because a species will vary. You will learn that with the Prof. Kanichi. Different species, one species will grow more in certain conditions. If you have a lot of crab exploitation, your crab will disappear. So fauna species are also indicate things about your forest condition. So you need to know what you want to know. And after that, what resources do you have? This kind of study is very expensive. You need a lot of time in the field, and you need, of course, expertise to collect the data and then to analyze the data and write the report. Do you really need all of this? So you need to bear in mind, what do you want? And then you decide what you want to do. Okay? More questions?
Do you need to inform management? Are you looking for a problem, or you want to find the cause of a problem? So most of your survey should have a focus, a goal, why you're going to do it.
Yeah. Who are you writing your report to is also very important. The way I would write a report for Prof. Amina is completely different from the way I would write the report for Mr. Philippe. He's a manager. He needs to know what to do. He doesn't care about
Size class.
no, that is nothing for him. So you also need to think about the audience. Am I writing for a science audience, for scientific audience, managers? Even managers and policymakers, the way they think is different. The policymaker will want, of course, he wants a little bit of management, yes, but he wants money, he wants votes, he wants profitability, he wants to make sure people are okay. Just people are okay. So he will say aquaculture, for example. The manager will say integrated aquaculture. The scientist will say biodiversity. You see? So at different levels, different expectations, you also need to think about that.
Sorry.
Yeah, we need a break.
It's like, I just understand that doing the work is one thing, and you also have to think who you're going to give to the information. It's like you can write one paper in 3 or 4 different ways.
Exactly, exactly. This study of the Zambezi Delta that we did, we wrote the report thinking that we are writing to a scientific. They said the work is very good, we like the graphs, and the tables, they look very good, but we don't understand. Please make it simple. So we made it simple. Then they gave us another feedback. Our policy makers, they also want to read the report and they understand nothing. And we had already made it simple. They understand nothing.
Yeah.
So for them, essentially, you just tell the story. You don't need to tell what the average DBH is. You just need to say, this place is being affected by this, and that, and this is what you need to do. So it's different ways. Yeah, we could write 3 reports, one scientific, one for managers, and one for the policymakers.
And one for my community.
Yes, if you also need to, it's very important. When you go and make a study, it's always good to find a way to bring feedback. Because especially if you do a socioeconomic study, people will be curious. They will want to know what was the result of the study. So you either make a poster and you send, sometimes you cannot go back. So you can make a poster and send to the administration, whatever, but always try to find a way to give them feedback. It's always good for them to know what is happening.
Yeah, because sometimes we forget about community, but they are the one making a lot of change to the environment.
Most of the time, the management interventions, they are the ones who are going to make. The policymakers, the authorities, yeah, they will be there, blah, 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 but who will talk the talk and do things is the community.
Yeah, but that approach is very important for us as a field officer, project manager, senior manager, because we work with
community. And we make a research with them. For example, in my project, we have to go with the community as an agreement. We have with the financial people. So we have to go with the community. So it's important for us, for me, to know that I can't give them the report as a scientific one, because when teacher was talking about that, I'm making one for my degree, of course, and I will need to report this to the community, and also the results we need to present to the decision makers about how they have to manage that.
Make sure you do that. You can send a poster to them. You do your study, then you make a poster, and you can send them a poster.